Honor of Dr. Caleb Finch, who is joining us today from USC, and he is a distinguished university professor there, one of only 27 at the university I counted. So we are very excited, and I realized that he's published over 600 papers because the paper he sent was labeled 610, and I thought, what is 610? And I realized, oh wow, that's impressive. So Dr. Finch will hear about some of his work. He studies the biology of human aging from an evolutionary perspective. And with that, I think we're just delighted to have you. Please join me in welcoming.
Well, I really enjoy the opportunity to talk with the anthropology community. I've known the molecular biology and biochemistry community for 50 years. And so my interest in the biology of aging is to understand how it interacts with the developmental process and with evolution. So this is something that everybody's aware of, but I would like to point out that Alzheimer's disease with neurodegeneration is found in a number of primates and even some of the shortest lived ones, but it has not been documented in the great apes. They have some level of cognitive impairment that would be equivalent to minimal cognitive impairment, or MCI, but there's been no neurodegenerative generation in the pathways that are specific for Alzheimer's. And that leads to some really interesting questions. So humans, this is a life history perspective, and it fascinates me that weaning is at age 2 typically in humans and under natural fertility, and it's 5+ years in the great apes. Puberty is not too different, but the age of first reproduction is, of course, later in humans. And menopause is at about the same, but great apes have lifespans that are relatively short post-reproductive, whereas humans added another 25 years. And we really don't know the mechanisms for that. And humans have vascular disease and Alzheimer's disease, which is absent in the great apes, and as I will show you, is absent in some human populations. So in the course of the evolution of humans, here is a paper 4 years ago. These are the gene changes in gene sequence and in gene expression, and I'm going to focus on ApoE3, which came into existence approximately in human populations at the time of the anatomically modern H. sapiens, and ApoE2 came in probably 100,000 years ago. And these have very different outcomes of aging and environmental interactions that I'll describe. But the point is that there's a number of different genes that came into existence at the time of different environmental exposures, which are described in The Evolution of the Human Exposome by Ben Trumbull and me. So desert dust was present in the earliest pre-Homo. Domestic fire probably as early as I'm hesitating because there is data saying that it was present 500,000 years ago, but that's controversial. But it really became established in human populations somewhere 100 to 50,000 years ago. And with that came exposure to eating charred meat on a regular basis and domestic smoke, which was treasured because it kept the bugs down in the huts that people lived in. And then only very recently, fossil fuels and tobacco. So here's the diagram for ApoE, and much more can be said, as I will tell you that ApoE is part of a gene complex that has variant alleles of different evolutionary antiquity in adjacent genes. So there's much to be worked out on the ApoE and evolution. But in modern populations, this is all populations of Denmark, all-cause mortality is highest for E44, which is on the bottom line in each one of these sets. Cardiovascular mortality is much higher. Dementia-associated mortality is even higher, but cancer mortality is not influenced by ApoE alleles in modern populations. And that's something to think about. So I want to start with showing some data from a project that I'm a collaborator on. The anthropologists Hilliard Kaplan and Mike Gervin began this project 20 years ago in the Tsimane in lowland Bolivia. And their population pyramid on the far right is the same as population of pyramid for Sweden 1750, with very high early mortality. If you got to age 25, you had another 40-year life expectancy, but essentially 10% of the population survived to age 60. Infections are more than 80% of the mortality causes, direct infections and septicemia from wounds, which are extremely common in hunter-foragers, farmers, because injury is extremely common. But there's no heart attacks. Less than 1% of mortality is associated with heart attacks, and there's no advanced Alzheimer's that I will show you. So these are personal photos of visiting villages in Bolivia with Kaplan and Gervin, and I will tell you in a moment my involvement with this, but everything is brown and sticky. You cannot get clean. And my anthropology colleagues commonly experience 3 days a month in which they have either lung or gut issues that prevent them from working, and that's about the same intrusion of acute bacterial and viral infections in the adult Tsimane. So they have high levels of blood CRP and interleukin-6. And I joined the project probably in 2012, and it's been an immensely interesting counterpart to my lab experimental research. So I was also working with a group of cardiologists who were interested in the whether cardiovascular disease was ancient or not. And I joined them when they were just beginning to image mummies, and those were some fascinating trips. And I told them about the Tsimane having no ischemic disease, and they said, well, that's impossible. Here, every mummy we have looked at has half have some coronary calcium. And so I got a team of first-rate cardiologists to come to Bolivia, and here is the first scan that we're doing. You can see some Chiman there and cardiologists. And the results were striking, that they do have coronary calcium at later ages, but their schedule of calcium increase is 20 years slower than North America and Western Europe. And there's a sex difference. Women have slightly less than men, so sex differences hold up. And then, of course, if the heart is good, what about the brain? And I got a team from our Alzheimer's group together, and Margie Gatz was the leader in this, wonderful scientist, and just published this year that the Chimon have the lowest prevalence of dementia worldwide. There's mild cognitive impairment, but no cases of advanced dementia. And of course, we don't have post-mortem data because that's not feasible within their value system of post-mortem sacredness. But so here is a population that I think can be used as a model for humans who evolved up through the Neolithic. And that leads to the possibility that cardiovascular disease and Alzheimer's disease on the scale that is known in all industrial societies are modern, and at a population level may be prevalent only at that level in the last 150 years. So this would be consistent with the great apes having no prevalence of either disease. So from that point of view, there is a change that is not related to natural selection in any way, but related in some way to the ecology that prevailed until very recently, that the Chimon and several other populations may have represented. So not only is the cognitive performance maintained better, but the rate of cerebral atrophy, here's another person I brought in who's a brilliant cognitive neuroimager, and the Chimon have a 20-30% slower cerebral cortex atrophy than North America and Western Europe. So aging in that level is slowed despite this small survival to advanced ages because of infection. So now I want to bring in the ApoE side of things, and I will give some surprising data, some of it very new, that ApoE4, the Alzheimer risk gene, is in fact protective under conditions of high infection. So paper that I was part of 6-7 years ago looked at the relationship of cognition and age versus evidence of infection by blood eosinophil count. And for these 6 different cognitive measures, the E3 and E4, E4 carrying had stable cognition better than the E3 did. So there seemed to be some protective effect. And then just a couple of weeks ago, we published this data that the ApoE4 has a benefit to reproduction, that carrying one E4 allele, there was 0.3 more births than someone who was E33, and 2 alleles, there was 2.1 more births, and that's the top curve versus age number of births. So in conditions of high infection, this is advantageous, and this extends findings from Ghana on another group that I was fortunate to be invited to join to discuss ApoE. And here is in villages, the survival of E4 carriers is, in the two right-hand curves, is better than E33 carriers, and the number of children that survive is greater in the villages with dirty drinking water. So there were side-by-side villages with clean well water, and they had higher childhood mortality. It turns out that the villagers preferred the river water because it tasted better. So to summarize this, past with high infectious deaths, life expectancy was under 45, small numbers, people getting later. Food was irregular. People did a much greater level of walking and working. In our world, us, chronic aging is the cause of most deaths, and we have available food in excess of our energy needs. And in the pre-industrial, the argument is that ApoE4 was adaptive. In our circumstance, it's negative. ApoE3, you remember, evolved about the time of H. sapiens, modern anatomical, and the ApoE3 has a major influence on synaptic density during brain development that E3 carriers during childhood and early adolescence have more synapses than E4. That there's about 4 or 5 papers on that now. So there may have been trade-offs on brain development and infectious vulnerability that has not been treated in depth, but it could be a wonderful set of inquiries. And just a gesture, I have always been interested in classic understanding Greco-Roman of aging, and I've written some papers on this. And it appears that in Greece, ancient Greece, there are many comments about slower cognition and slower learning, but none on anything describing advanced dementia. And in Rome, 400 years later, there are at least 8, and that's the subject of the paper that's under review, accounts of advanced cognitive impairment that would superficially be consistent with Alzheimer's. So what's the difference between ancient Greece and Rome? Well, there is one that came to light from a study that I did with a terrific classics scholar, Stan Burstein, on lead, and it turns out that the Romans liked to add lead acetate to their wines because it made the wine sweeter. And there's a small literature that supports lead exposure as a risk factor in Alzheimer's. More to that I can answer. So now I want to look a little bit into the ApoE story. And it turns out that in all mammals, well, let's just say mice and humans, that ApoE is part of a gene cluster that travels together in the same order. And this is 150 million years of elapsed evolutionary time. Same order in mice as in humans. It happens to be translocated in reverse. And the point is that there are a number of other genes that are adjacent to ApoE that regulate immunity, metabolism, and reproduction. And there are, they modify the impact of the ApoE4 allele by ethnic background. So the E4, E3, and E2 have different haplotypes in evolution that reflect their history and selection in ways that have not been decoded. And in some populations, ApoE4 is not a risk factor. It's much less of an Alzheimer's risk factor in West African populations, Yoruba particularly, but there's a whole wonderful opportunity to follow this out with Bantu speakers. So I consider that the ApoE gene, ApoE4 effects, all of them have to be revised and re-looked at and reanalyzed for the haplotypes of the adjacent genes that modify the impact of ApoE4 on outcomes of aging. And I've considered, written some articles about ApoE as a part of a life history gene complex because of its influence on immunity, growth, brain development, and aging. And just a glimpse of some of the genes that are next to it, covering all of this aspects of physiology. And we'll get to it in a moment, but it turns out that the mice with ApoE, human ApoE3 and ApoE4 have different responses to air pollution in the genes that are activated. So putting all this together, to organize my own thoughts, I borrowed a concept from the cancer literature of the exposome. A single carcinogen is active in an ecological context. And so we have exogenous macro level differences between individuals, diet, lifestyle, exposure to air pollution, cigarettes, and that are an exogenous individual and then endogenous individual. And one of those endogenous individual factors is the amount of body fat that is an environmental interaction and influences heart disease and outcomes of brain aging. So it's useful. It's a word, an exposome, so I don't want to make more of it, but it gives a framework for thinking of the complexity of these interactions. So now genetics. As I tell my undergraduates when I give this lecture, if you have the genes of a mouse, you'll live 2 years. If you have the genes of a human, well, that's up to you to some extent. And it's been a remarkable observation that turned up in this review that I wrote, well, was it 1997, 25 years ago, that the heritability of lifespans in identical twins is rather low. It's 30%. And most recently, if you look at the heritability of identical versus non-identical at age 80, it's about 10%. So this says that environmental factors, including lifestyle, may profoundly influence outcomes. And that has led me to studies of air pollution for reasons that we'll get to. But the point is that diet, air pollution, infections, social stress all feed into the major diseases of aging. And this was the precursor to the exposome. And I just want to emphasize the shared commonality of cardiovascular lesions and Alzheimer lesions. They both involve the same monocytic cell type, macrophage, foam cell in the atheroma, and microglia in the brain. And if you compare the inflammatory components of atheroma and senile plaques, they have a huge overlap. So here we have a basis for thinking that systemic inflammation engages both the heart and the brain, and it turns out that all of the risk factors for Alzheimer's, or almost all of them, are shared with risk factors for cardiovascular disease. And that's something that my neuroscience colleagues have been very slow to assimilate, because the brain ends at the neck, right? So at the same time that I was thinking about this, my colleagues in epidemiology at USC and UCLA were showing how air particle density in the LA basin was associated with degrees of cognitive impairment. And here is a map, and where USC and UCLA are in the brownest zone. And the reality is, for home-dwelling older people, that each of the components listed here of air pollution had a stronger association with one group of cognitive processes that are changed with aging. These are not demented, these are interacting with normal aging. So I've been developing an experimental approach to this with J.C. Chen, who's an epidemiologist, and at the same time I was doing some experimental studies that I'll describe. He launched a really deep set of epidemiological studies, well, not epidemiological, but cohort-based studies with the Women's Health Mental Initiative. And there's 15 papers which essentially draw the conclusion that exposure to air pollution accelerates gray matter and white matter loss with some specificity for the regions that are accelerated in Alzheimer's disease. And there's essentially a 10 to 15%, I have a slide coming up that says this, of air pollution, of dementia can be associated with exposure to high levels of air pollution, as well as cigarette smoke. So had an important other collaboration with Kostas Sayoutis, who's in the engineering school, who devised machinery for instruments for collecting air pollution particles and re-aerosolizing them for exposure of mice to precise time and dose of air pollution particles. And this has been highly productive. We have a program project on this. And among the other things that I'll describe, mice carrying human ApoE4 genes have more amyloid than the E3 in exposure to air pollution. And we have quite a story on how that works up, and I won't go through it in any detail. But it turns out that some human populations also show greater vulnerability to air pollution components in consecutive visits in the Northern Manhattan Cognitive Study. You can see that the blue curve, which is E4 containers have faster slopes for nitric oxide, PM 2.5 or PM 10. So this is an example, a putative example still being worked out of gene-environment interactions in response to air pollution, and much more to be learned about that. So we've done pretty detailed studies of gene responses by ApoE allele, and there are sex differences in the genes that respond, and we've got a set of pathways that were defined here that are fundamentally come down to influences on transcription factors, NF-kappa B and NRF2, that regulate batteries of genes for antioxidant repair and that are relevant to Alzheimer's disease and also brain development, I think. So almost everything that air pollution does, cigarette smoking does. And so we compared the gene responses to air pollution with genotypes in humans found by Eileen Crimmins that are protective for survival of lifetime smoking. And it turns out that the bolded genes respond to air pollution in mice, and those are the same genes that have variants that also are protective for lifetime smokers reaching the age of 80. So now I want to shift the last phase of it towards development. And here's another study from USC, Rob McConnell's group, looking at childhood obesity, well, not child, 10 to 18, in people in Los Angeles, kids in Los Angeles. The bottom line is who live near a major roadway. The second blue is live in a household where there are adult smokers, and both of those increase the BMI. And then the combination of secondhand smoke and near roadway is super additive. So there's some toxicology here on the basis for super additivity that is, I'll just say it out, I don't have a slide, documented for interactions of cigarette smoke and air pollution for lung cancer. And even though ApoE is not identified as a lung cancer gene, ApoE alleles influence response to cigarette smoke and to air pollution. So there may be a really interesting epidemiological matrix which will map into transcription factor responses of genes that deal with antioxidant repair. So the last phase, I want to turn to socioeconomic status, which is studied by a close colleague, Eileen Crimmins, and she has a paper 15 years ago that my biology colleagues are unaware of, that there are 10 to 15 years lifespan differences between low education, low SES, and the highest group. And it turns out, as I will show you, there's a parallel difference in the onset of the major diseases, cardiovascular, and here is shown for Alzheimer's. By income decile, there is a linear relationship to the risk of dementia. And the linearity of this is observed across SES in a number of different studies. And I don't really understand how anything this complex could be linear, but it turns up in most studies. So children from low SES families are born biologically disadvantaged. There's impaired brain development in the frontal cortex and in the hippocampus. As children, low SES have worse executive function, more childhood obesity, and maternal smoking has something to do with this, and maternal smoking influences DNA methylation of the unborn fetus. So a lot of biology here. So 2 days ago, I happened to discover a wonderful set of studies called SALSA, Sacramento Area Latino Studies on Aging, that's been, I knew about it because I had some role in helping it get started 20 years ago when I was still active in the Alzheimer's program. And so what they did was to examine cognitive function at later ages depending upon high-low childhood, high-low early adulthood, and high-low later life. And the two extreme curves are a survival curve of cognitive function in the triangles here are low, and the black dots are high. So this just shows, again, the SES component. And then this study also showed that those who went from low to higher SES during adult life retained the disadvantage in terms of their higher risk of dementia or cognitive loss. So we're looking at this for developmental exposure to air pollution, which on the average is greater in lower income households because they can't afford to, the land apartments are closer to freeways and there's more adult smokers. And what we've found, and I'll show some of the data, that exposure of mice in gestation only alters their adult physiology in their brain. So here is an example of the brain function. This is adult neurogenesis in the dentate gyrus from the stem cells that we all have that continue to add hippocampal neurons. And here is the black bar shows the impact of developmental exposure to air pollution. So it's more than 50% reduced in the adult. And we've replicated this in 2 other studies, so I'm pretty confident about it. And then the adult mice tend to be fatter. They tend to have poor glucose tolerance. And their hypothalamic peptides, by this one single observation, look altered. So 3 out of 5 are lower in the as adults in the hypothalamic peptides for those who are gestationally exposed. So this is, I think, gives us a basis for a larger discussion of framework, in a framework comparing the low SES, which is in the horizontal bar, shows earlier mortality and obesity, shorter healthy adult lifespan, earlier CVD and Alzheimer's. And there's data from adoption studies that is incompletely analyzed, it's published, but that gives some insight that kids adopted from high to low and low to high as adults carry over their earliest phase, whether it's as neonate adoption or early childhood adoption. And then the data I just showed from SALSA suggests that there's carryover in the adult transition. So there's an immense amount of work to be done on this, and I'm trying to learn how to think about the biology of SES, which is something that has been often attributed to just social practice. But there's a deeper biology here. And it very much goes with Molly's findings on input from grandparents on cortisol levels. So there's a potential integration here between our labs. So outcome of this is future of healthy aging, brain and otherwise, is how to minimize gerogens, air pollution, bad diet, etc., and to optimize gene by environment interactions, development through aging. And I would like to end with one biological fact, that the egg we came from was in our mother's ovary at the time of her birth. That's absolutely clear developmental biology, which means that we are potentially exposed, carrying over grand maternal environmental effects, which in my case, my grandmother was born in 1878. Open for questions.